Hello everyone. In this video we are going to discuss about lac operon. Okay. So before understanding what is lac operon, you should first understand what is an operon. So what are operons? Operons are units of genetic material in prokaryotic organisms such as bacteria, consisting of a cluster of genes under the control of a single promoter. We are considering a prokaryotic genome. Say there are we are considering three genes now: gene 1, gene 2, and gene 3. Okay, these three genes are seen adjacent to each other. Okay, and if transcription has to happen, there should be the presence of a promoter where RNA polymerase comes and binds. And the binding of RNA polymerase is basically going to initiate the process of transcription. Okay, now the concept of operon is that this single promoter over here is controlling the expression of gene 1, gene 2 and gene 3, meaning this single promoter is going to control the expression of these three genes. Okay, so basically transcription will start from here and gene 1 will get expressed and gene 2 will get expressed and gene 3 will get expressed. Gene 2 and gene 3, they don't have separate promoters. They both the three all the three genes, gene 1, gene 2, gene 3, they are dependent on only one promoter, or they have only one promoter. That is the concept of operons. So operons are like the cluster of genes under the control of a single promoter. That is operon. So before understanding lac operon, you need to understand some terms such as activators, activators. Okay, imagine a promoter. Consider a operon. Okay, there is a promoter and this promoter is controlling the expression of three genes: gene 1, gene 2 and gene 3. Okay, and all these genes codes for different enzymes. Okay, different mRNAs, which ultimately codes for different proteins. Okay, my question to you is that will these three genes be expressed all the time, or will it be expressed only when there is a need? So if there is a need of these three proteins always, it will be expressed always, else it will be expressed only when it has to be. Okay, that now we are coming into the concept of activators. Okay, so if this three genes has to be expressed only when it is needed, then there should be something which is controlling the expression of these three genes, and that is basically our promoter. RNA polymerase is going to come and bind to a promoter region, and that is something which is making this happen. So if the RNA polymerase does not bind with the promoter region, the expression of these three genes will not happen. So if there is no need of expression, then what is going to happen is there will be something bind to this promoter. Okay, there will be something bind to this promoter. By the way, I'm explaining about repressor right now. Okay, activators we'll discuss after repressor. So uh, so if RNA polymerase is not being able to bind to the promoter, then the expression of gene will not happen. So imagine this is my promoter site, and here the RNA polymerase comes and attaches. Okay, but I don't want that to happen. The cell doesn't want that to happen because there is no need of it at that time. So a repressor molecule, a compound called repressor, is going to bind to the promoter, and the site is no longer available for the RNA pol enzyme, RNA polymerase enzyme, and this is called a repressor. So now what is an activator? An activator is something which will go and bind to the promoter region. Okay, it will go and bind to the promoter region and leads to some conformational changes in the promoter region. Conformational changes in the promoter region. Conformation is the arrangement in space. Okay, so it leads to some conformational changes in the promoter region and make it easy for RNA polymerase to come and bind to it. So that promoter region was not available for the RNA polymerase. So what the activator did was it activated that promoter site. It activated the promoter site and made it accessible to the RNA polymerase. So that is basically an activator. So an activator activates the gene expression, and a repressor represses the gene expression. And okay, where will this repressor be present? This repressor will be present in a gene which is in always on condition. Sorry, an operon which is in always on condition. Always on condition in this sense, if this repressor is not bound to this promoter region, transcription will happen, keep on happening. So there is no stop. Okay, if this repressor is not there, definitely RNA polymerase enzyme is going to come and attach to this site, and transcription will continuously happen even when there is no need. That is when our repressor comes into play. This repressor will come and bind to that particular site and will block the transcription process from will block the RNA polymerase and prevents the transcription process from happening. In the case of activator, what happens is uh, this gene is no this gene is sorry this operon is not activated, so the promoter site is not available for the RNA polymerase. So it requires something to make a conformational change so that RNA polymerase will be able to access the promoter site. So that is an activator. This is in the case of a gene which is in always off condition. This gene will be always off. If there is no activator, no expression of this gene is going to happen. So now let us go to our lac operon. So this is our lac operon. Okay. See, the lac operon controls three genes Z, Y, and A. This Z gene codes for an enzyme called beta galactosidase. Beta galactosidase, and this beta galactosidase it converts lactose to glucose plus galactose. Okay, this beta galactosidase converts lactose to glucose and galactose. So when there is a presence of lactose, so lactose will not be present in cell always. So if there is a presence of lactose, this Z gene has to be expressed. That is what the cell wants. So why it codes for an enzyme called permease. So permease basically helps in the transfer of lactose across the cell membrane. Okay, the transport of lactose. And A, it codes for transacetylase, an enzyme called transacetylase. Now you could see a promoter region, an operator region. So this promoter region is the one which controls the expression of these three genes Z, Y, and A. This is where RNA polymerase binds. So what is the function of operator site? It basically operates the function of these genes. Operator site is the site where the activator comes and binds, and here the conformational change is going to happen, and RNA pole will come and bind to this site, this the promoter site and operator site, and it will kickstart transcription. So you have seen three genes Z, Y, and A. Now, my question to you is that do we need this gene to be expressed all the time? No, because cell considers sorry, glucose as the main source of energy. The cell will utilize lactose if and only if glucose is not present. Okay, imagine you prefer coffee to tea. You will drink tea if and only if coffee is not there in the show. Okay. Likewise, the likewise what's going to happen here is if glucose is not present and lactose is present. Okay, glucose is not present and lactose is present. Now in this case what is going to happen is there is a repressor attached to this site. Okay, and this is the gene which codes for that repressor, and this is its promoter region. So this gene produced a repressor mRNA, and this repressor mRNA got translated and it became a repressor protein. This repressor mRNA is converted into repressor protein, and this repressor protein has gone and bind to the operator site. Now the operator site is not being able to let RNA polymerase access its site. So my question to you is whether this is a always on operon or always off operon. This is an always on operon, because if the repressor is not there, this will be in always on condition. Okay, now the repressor comes and binds to this site, and it basically blocked the RNA polymerase from accessing the operator site. Now the gene is no longer in always on condition. This is the case when glucose is present or lactose is not present. Okay, glucose is present. This is the condition when glucose is present or lactose is not present. So if glucose is present, even if lactose is there, this gene will be in repressed state. I will tell you the reason shortly. So this repressor gene will go and bind to the operator site. This is the case when there is no lactose. Now glucose is not there in the cell and lactose has entered the cell. Okay, this is our cell. Now lactose has entered the cell. Now this repressor this repressor protein bound to the operator region has to be removed. And how will it get removed? As soon as the lactose enters the cell. And what helps in the entry of lactose? Permease. So I have said that the gene Y is not getting expressed and gene Y produces permease which lets lactose enter the cell. But if Y is not if the expression of Y is inhibited, now what is producing permease? You'll be having this question in mind. So the answer is, even if the repressor is bound to the operator side, this binding is not 100% efficient. Okay, this binding attaches and detaches at a regular interval. Okay, when this get detached, a small amount of Y gene will be transcribed. This is called leaky transcription. This is called leaky transcription. Okay, so as a result, very small amount of permease will be produced, and that lets the entry of lactose into the cell. Okay, as soon as the lactose enters the cell, it will be converted to allolactose. Okay. It will be converted to allolactose. So okay, as soon as the lactose enters the cell, it will be converted to allolactose, and this allolactose, what it is going to do is, this allolactose will go and bind to the operator region. Sorry, it will go and bind to the repressor in the operator region. When the allolactose binds to the repressor, the repressor will become inactive, and it will come out of the operator site, and the repressor will be repressed now. So the repressor will no longer be active. Okay, and this conversion of lactose to allolactose is done by beta galactosidase enzyme. Okay, and this allolactose goes and binds to the repressor, and now the gene is open for RNA polymerase. Now the RNA polymerase will come and bind to this site, and transcription happens. Z gene will be transcribed, Y gene will be transcribed, A gene will be transcribed, and they will be converted to corresponding proteins. Okay, this is the repression is what is shown here. So the repressor is here. Okay, the allolactose comes and bind to repressor mRNA repressor. Okay, now the allolactose will come and bind to the repressor, and the repressor will be inactivated. Now I told you that the expression of the lac operon will happen only if the glucose is not there. The reason for that is, when glucose is present, what is going to happen is, for transcription to happen properly, a C A M P C B P complex has to go and bind to the operator region. Only then the transcription of the gene will be happening properly. So, and how does glucose affect this? So there is a compound called adenylate cyclase, which is a precursor of CAMP. Adenylate cyclase, it is a precursor of CAMP. Okay, but when glucose is present, glucose will inhibit the conversion of adenylate cyclase to CAMP. So as a result, when glucose is present, even though lactose is there in the medium, it will not be the operator site will not undergo any conformational change. The RNA polymerase will not be able to bind to the operator site properly, and transcription will not happen properly. This is the reason why, if glucose is there in the medium, it will act as an inhibitor for the inhibitor for these three genes. It will block the it will not let the operator site to get activated. Okay, that is the reason why this will the expression of this gene will happen if and only if only lactose is present in the medium. Okay, there shouldn't be any glucose. So that's it, we are done. Thank you for listening to my lecture. I hope you have enjoyed it. Bye. Bye bye.